if you've ever taken a prescription for like severe acne or maybe a stubborn urinary tract infection or even Lyme disease, there is a very good chance you swallowed a tetracycline.
Oh, absolutely. I mean, doctors and microbiologists have been relying on these antibiotics since the 1940s.
Right, which is wild to think about. We've prescribed them, what, hundreds of millions of times? And for the last 70 years, we basically thought we knew exactly how they worked.
We did, but a new structural biology paper just proved that we've actually been missing half the story this entire time.
Which is just mind-blowing. It forces us to completely rethink, a fundamental piece of modern medicine.
Yeah, what we're seeing now is that these ubiquitous everyday drugs have been operating with this hidden secondary attack mode right under our noses.
So today we are taking a deep dive into that discovery. We're looking at a brand new paper titled Dual Site Targeting of the Bacterial 7DS Ribosome by Tetracyclines.
It's a fascinating paper.
It really is. And our mission today is to explore how these familiar drugs pull off this hidden dual-site attack at the atomic level, which is thanks to revelations from high-resolution cryo-electron microscopy.
Right. And more importantly, we want to look at how mapping out this secondary attack mode might actually give us the blueprint we need to build new drugs.
Yeah, drugs that can hopefully overcome the global crisis of antibiotic resistance. So, okay, let's unpack this. To really understand the magnitude of this structural plot twist, we have to look at the established dogma first.
Right, the baseline of what we thought we knew.
Exactly. So we've always known that tetracyclines target the bacterial ribosome, right? Like the cell's protein-making machinery. And they specifically do this by parking themselves in the 30S decoding center to block incoming transfer RNAs.
Yeah, that was the classic mechanism. The drug binds to what we call the canonical binding site.
The canonical binding site.
Right.
Yeah.
By sitting there on the 30S subunit, it physically obstructs the raw materials from entering the assembly line.
So it's basically jamming the gears.
Exactly. The bacteria can't build new proteins, so it stops growing and eventually dies. It was a very clean, established mechanism that, honestly, no one really questioned.
But the new cryo-EM images of modern tetracyclines, and we're talking about drugs like minocycline, doxycycline, sarecycline, they reveal they aren't just jamming that incoming assembly line.
No, they're not. They are simultaneously attacking the 50S subunit.
Okay, wait. The 50S subunit, what is happening there exactly?
So specifically, they're targeting a region inside the 50S subunit called the nascent peptide exit tunnel.
Okay, exit tunnel, okay.
Right. To visualize this, whenever the ribosome successfully links amino acids together to build a new protein chain, that growing chain has to leave the machinery through this specific exit tunnel.
Oh, I see.
And what the researchers discovered is a secondary binding site right inside that tunnel.
Okay, so if I'm picturing this for a second, if the bacterial ribosome is like a 3D printer, the classic view said that the antibiotic works by unplugging the data cable, right? The 30S subunit.
Yeah, the data cable analogy is perfect.
So the printer just stops receiving instructions on what to make. But this new data shows the drug is also like aggressively shoving a cork right into the nozzle where the printed plastic is supposed to come out.
Yes, that analogy holds up perfectly. You're cutting off the instructions and you're physically plugging the exhaust.
That is nuts.
It is. The drug anchors itself right next to the active center where the amino acids are being linked. So it creates a literal roadblock for the newly forming protein.
Okay, I understand how it works mechanically, but I'm really struggling with the timeline here. Like, we have been studying tetracyclines using X-ray crystallography for decades.
Decades, yeah.
So how did countless scientists miss a massive structural roadblock sitting right inside the exit tunnel?
It actually comes down to the physical limitations of how we used to image these molecules.
What do you mean?
Well, in traditional X-ray crystallography, researchers often had to grow solid, rigid crystals of the bacterial ribosome first. And once they had a stable crystal, they would try to basically soak the antibiotic into it.
So they're pushing the drug into a solid crystal.
Right. But in a tightly packed crystal lattice, the exit tunnel is often physically blocked or restricted. Yeah. So the drug simply couldn't navigate deep into the tunnel of a preformed crystal.
Oh, wow. So the methodology itself was hiding the truth. Because the crystal was too dense, the drug only bound to the easily accessible 30S site on the surface.
Exactly. Which, of course, just reinforced the existing dogma. We saw what we expected to see.
That is wild.
But then cryo-electron microscopy, or cryo-EM, changed the game.
Right, because cryo-EM doesn't use crystals.
Exactly. Instead of growing rigid crystals, you flash freeze the molecules in a much more natural liquid state.
Oh, so they're floating around naturally.
Yes. The ribosomes and the drugs are interacting in a solution just as they would inside a living cell.
So the tunnel is open.
Precisely. Because the tunnels were open and accessible before freezing, researchers could finally see the drug happily binding to this hidden secondary site.
So just to recap a bit for you listening, we've learned that for 70 years, our testing methods essentially locked the door to the exit tunnel, hiding this massive secondary attack mechanism from science.
Yeah, that's exactly what happened.
But now that we can see it, I noticed looking through the sources that the physical mechanism of how it plugs the tunnel is, well, it's incredibly dynamic.
Oh, very much so.
Like the drug doesn't just jam itself in there the exact same way every time, right?
Yeah.
Depending on the specific bacteria, the drug has to perform some intense atomic gymnastics to make it fit.
The precision here is truly remarkable. To really appreciate it, you have to picture the core chemical structure of a tetracycline molecule.
Okay.
It consists of 4 continuous rings. They're labeled A, B, C, and D.
A, B, C, D.
Got it. Right. And they're arranged a bit like a rigid molecular caterpillar. When this structure enters the bacterial exit tunnel, it's basically searching for a very specific structural landing pad in the bacterial RNA.
And that landing pad changes depending on the pathogen, right?
Yes, exactly.
For instance, in Cutibacterium acnes, which is the bacteria that causes severe acne, the tunnel has a landing pad made of a specific pair of cytosine molecules, a CC pair.
Right. That cytosine pairing creates a very specific physical and electrostatic environment. So the tetracycline molecule slides in, and its middle rings stack perfectly perpendicular against those cytosines.
Wow, okay.
And to ensure it doesn't just wash away, the drug coordinates with nearby magnesium ions. It essentially uses them as chemical anchors to lock itself tightly to the walls of the tunnel.
That makes sense. But then you look at an enteric bacteria like E. coli, which is responsible for things like food poisoning or UTIs.
Right.
The structure of the E. coli tunnel is slightly different. Instead of that cytosine pair, it has a uracil pair, a UU pair.
Yeah, and that one substitution, literally just swapping cytosines for uracils, completely changes the internal landscape of the tunnel.
It's crazy that such a tiny change matters so much. Because of that change in the terrain, the drug has to contort itself.
It does.
Like, to successfully block the E. coli tunnel, a drug like doxycycline has to rotate roughly 90 degrees along its axis compared to how it sits inside the acne bacteria.
Yeah, it has to turn completely sideways just to wedge itself in there.
But that rotation, I mean, that's significant, but it's nothing compared to the structural gymnastics required from some of the newer drugs, right?
Oh, you mean like sarecycline?
Yes, sarecycline. It's a third-generation tetracycline.
Right, so medicinal chemists actually designed sarecycline with this bulky structural extension on one of its rings, specifically to help it overcome certain types of bacterial resistance.
Okay, wait, let me pause you there. If sarecycline has a bulky arm and it has to rotate to deal with that uracil pairing in E. coli, wouldn't that make it a terrible antibiotic for that specific bug?
Why do you say that?
Well, wouldn't that bulky extension just smash into the wall of the tunnel and, like, bounce off?
You've hit on the exact structural problem the drug faces. If it tries to enter the E. coli tunnel the normal way, that bulky extension physically clashes with the tunnel wall.
Oh, so it just doesn't fit.
Normally, no, it doesn't. But the cryo-EM images reveal this fascinating solution. To avoid that clash, sarecycline literally flips itself completely upside down.
Like a full 180 degrees.
A full 180 degrees. It reorients its bulky extension into an entirely different pocket of the tunnel just to secure a fit.
That is just a highly dynamic molecular dance just to achieve a blockade. And for you listening, I want to emphasize why this atomic choreography is so important.
Yeah, it's crucial.
These tiny twists and flips actually dictate drug efficacy. If an antibiotic can't successfully contort itself to fit the specific tunnel of the bacteria making you sick, it won't plug the exit and the bacteria might survive.
Exactly, which is why understanding these structures is so incredibly vital for the future of medicine. We're really moving past the point of just treating all bacteria as identical targets.
Right. Okay, so that brings us to one specific drug that doesn't just adapt to the tunnel, it completely overwhelms it.
Doxycycline.
Doxycycline. It's one of the most commonly prescribed antibiotics out there. But while all the tetracyclines we've discussed bind to this exit tunnel, doxycycline has a unique mechanism that the others just don't seem to replicate.
Right, it forms pairs, or what chemists call dimers.
Dimers.
So instead of entering the secondary binding site as just a solitary molecule, doxycycline molecules actually team up to build massive multi-drug roadblocks at multiple locations inside the exit tunnel.
The architecture of these blockades is just fascinating. The researchers identified 2 distinct doxycycline dimers at work here, right?
Yes, they did.
Let's look at the first one, which they named DoxD1. So we already have that single doxycycline molecule anchored in the secondary binding site we just talked about. But then a doxycycline dimer comes along and stacks directly on top of it.
Right, creating a trimer, a 3-molecule stack.
A 3-molecule stack inside the tunnel.
And location is everything here. This stack forms right next to the active center where the new protein is being synthesized.
Oh, wow.
When you look at the mapping, this 3-drug structure basically creates a physical ceiling.
So as the bacteria tries to thread a newly formed chain of amino acids down the tunnel,
the chain immediately slams into this massive doxycycline barrier.
it just completely halts production.
Completely.
But the drug doesn't stop there, because further down the exit tunnel, closer to where the protein would normally emerge into the cell, there is a second dimer blockade.
Right, DoxD.
DoxD2. And this one interacts with the tunnel walls in a way that fundamentally disables the bacteria's downstream processes.
It really does. So the walls of this tunnel are lined with crucial ribosomal proteins, specifically 2 known as UL22 and UL23.
Okay, UL22 and UL23.
And these aren't just for structural support, they have active jobs. The UL22 protein has a loop that helps fold new proteins into their correct 3-dimensional shapes as they exit.
Okay.
And the UL23 protein acts as a biological sensor. It basically identifies certain proteins and helps direct them to the cell membrane.
So just to simplify, UL22 is the folder, ensuring the protein actually works, and UL23 is like the shipping department, making sure it goes to the right place in the cell.
I love that, yes, the folder in the shipping department. And the DoxD2 dimer physically wedges itself right into the structural loops of both proteins simultaneously.
At the same time.
At the exact same time. It binds to them, completely restricting their movement and function, so it shuts down the folding mechanism and blinds the shipping department all at once.
That is just a brutal attack on the bacteria. And if we look closer at that shipping department protein, the UL23, we actually find an atomic level explanation for a well-known medical reality regarding Lyme disease, don't we?
We do.
Because doxycycline has always been the gold standard treatment for Lyme, which is caused by a tick-borne bacterium called Borrelia burgdorferi.
Right.
But the chemical reason why doxycycline is so uniquely devastating to this specific bacteria hasn't always been completely clear to us.
No, it hasn't. But the structural mapping provides just a beautiful answer to that. When you look at the genetic sequence of the Lyme bacteria, its UL23 protein is slightly different from most other pathogens.
How so?
Well, it has an extended structural loop that reaches further out into the exit tunnel.
Oh, and that extended loop happens to be the exact perfect shape to cradle the DoxD2 dimer.
Exactly. The dimer locks onto the exact chemical structure of that extended loop, binding to it with just incredible affinity.
So the Lyme bacteria's own unique structural quirk inadvertently creates a custom-made docking station for doxycycline.
Yes, it's a perfect fit, which totally explains why the drug binds so strongly and is so effective at clearing tick-borne infections compared to other tetracyclines.
Okay, so a quick recap before we move on. E. coli requires a 90-degree rotation. Sarecycline has to flip completely upside down, and doxycycline is forming massive multi-drug roadblocks that perfectly exploit the unique biology of things like Lyme disease.
That's the summary, yeah.
But as fascinating as these blockades and custom fits are, we really have to look at the practical biology.
Right, the clinical side.
Because seeing a drug form massive trimers and dimers in a highly controlled, purified liquid under an electron microscope is one thing. The real question is, does this secondary attack mode actually happen inside a human body?
And that is the crucial hurdle for any structural biology paper. I mean, if a drug only forms these roadblocks at insanely high concentrations that would be toxic to a patient, then the discovery, while academically interesting, has no clinical relevance.
It's just a lab trick at that point.
Exactly. So the researchers address this by analyzing the binding affinity, basically how strongly the drug is attracted to these different sites.
And they found that the secondary site inside the exit tunnel has a lower affinity than the primary site on the 30S subunit, right?
Right, meaning the primary site is much more attractive to the drug. At low concentrations, the drug acts exactly as the classic dogma dictates. It all flocks to the 30S decoding center. But as the concentration of the drug increases, that primary site gets fully saturated. The researchers discovered that once the drug concentration crosses a specific threshold, which is around 200 micromolar spines, the drug floods into the secondary site.
And that's when those massive doxycycline dimers fully engage.
Yes, exactly.
So the 200 micromolar threshold is the key. Let's relate this to a standard patient experience. If you swallow a normal oral doxycycline pill, the concentration of the drug in your blood plasma peaks well below that threshold, doesn't it?
It does. It doesn't trigger the secondary attack mode in your bloodstream.
But the human body doesn't distribute drugs evenly?
No, it doesn't. And that's the key. Doxycycline is cleared from the body through the kidneys. As the kidneys filter the drug out of your blood, they heavily concentrate it into your urine.
Oh, so your bloodstream might not hit the threshold, but your bladder becomes the perfect environment for this dual attack.
Exactly. The concentration in urine skyrockets well past the required threshold.
Which perfectly explains the clinical reality of why doxycycline is so effective at clearing out urinary tract infections.
Yes. In that specific local environment, the secondary site and the dimerization completely overwhelm the bacteria.
Wow. And I'm guessing it's the same principle for topical treatments, right?
Yeah.
Like, if you use a concentrated minocycline foam directly on your face for severe acne, the local concentration on your skin is going to be orders of magnitude higher than the threshold.
Absolutely. The local tissue environment easily supports this hidden secondary mechanism.
That makes total sense.
And this secondary mechanism isn't just an interesting quirk of high concentrations. It actually serves as a built-in weapon against antibiotic resistance.
Wait, really? How did they prove that?
Well, the researchers ran tests using engineered strains of E. coli to prove this.
Right. I read about this. They used bacteria that had evolved specific mutations to alter the physical shape of their primary 30S binding site.
Yeah, essentially the bacteria built a shield. Under normal, low-concentration conditions, these mutant bacteria are totally immune to tetracyclines.
Because the drug can't dock at the primary site, so the bacteria just survives and keeps replicating.
Right. But when the researchers exposed these immune bacteria to high concentrations of doxycycline and minocycline, the bacteria died anyway.
Which seems completely counterintuitive. If the primary target is mutated and shielded, how is the drug still killing them?
The exit tunnel.
Oh, of course.
Because the primary site was blocked, the high concentration of the drug just flooded directly into the secondary binding site in the tunnel, forming those dimer roadblocks.
So the secondary site essentially acts as a built-in atomic backup mechanism.
Exactly. Even if the bacteria manages to mutate its primary vulnerability, a high enough local concentration of the drug allows it to completely bypass that resistance.
That is incredible, though I do want to point out for the listeners, not all the drugs pass this specific mutant test.
That's true. Tetracycline struggled here.
Right. Because it has that bulky structural arm we talked about earlier, it couldn't easily maneuver into the secondary site in E. coli. Its backup mechanism just wasn't strong enough for this specific pathogen.
Which really underscores exactly why mapping these structures at the atomic level is so critical.
Right, because it gives us a visual guide to what modifications help a drug bypass resistance and what modifications hinder it, depending on the specific species of bacteria.
Precisely. And that brings us to the ultimate implication of this research. Now that we have this incredibly detailed atomic map of the dual site mechanism, how does this actually help medicinal chemists build better drugs?
That is the million-dollar question.
Well, it basically replaces trial and error with rational design. For decades, chemists would just make slight modifications to the tetracycline core and then test it to see if it worked better.
But without always knowing why it worked better.
Exactly. But now we have a literal blueprint of the bacterial exit tunnel.
We can see the exact shape of the pocket we're trying to plug.
And crucially, we also know the shape of the pockets we want to avoid.
Oh, that's interesting. What do you mean by pockets we want to avoid?
Well, the researchers highlighted this by looking at another related antibiotic, tigecycline.
Okay, tigecycline.
So tigecycline is highly effective against bacteria, but it have a known side effect. It can actually be toxic to humans.
Wait, why is it toxic to us?
Because it inadvertently binds to the secondary site inside human mitochondrial ribosomes.
Oh, wow. So it's catching our own cellular machinery in the crossfire.
Exactly. But the structural map shows us the mechanism behind that crossfire. Tigecycline has a specific structural extension on one of its rings. When it enters a human mitochondrial ribosome, that extension reaches deep into the active center, causing a physical clash that harms the human cell.
Okay, I see where this is going. So if we know the exact chemical extension that causes the clash in humans, and we know exactly how the human exit tunnel differs from the bacterial exit tunnel,
Chemists can engineer a way around it.
we can essentially edit the drug.
Yes. By adding highly specific structural modifications to certain positions on the tetracycline core, scientists can design new drugs that perfectly fit and jam the bacterial ribosome, but physically bounce off the human ribosome.
That's brilliant. We can systematically engineer the human toxicity out of the drug while maximizing its ability to build roadblocks inside bacteria.
Right. And it also opens the door to fundamentally changing how we approach antibiotic prescriptions in general.
You mean moving away from broad-spectrum antibiotics?
Exactly. Currently, when you take a broad-spectrum antibiotic, it's a systemic sweep. It wipes out the infection, but it also decimates the healthy, necessary bacteria in your gut microbiome.
Because the broad-spectrum drug is targeting features shared by almost all bacteria.
Right, but this new mapping shows us that the exit tunnels are not identical across all species.
Which we saw today. We just discussed how enteric bacteria like E. coli have a specific uracil pairing in their tunnel.
Right.
And we know acne bacteria rely on a cytosine pairing. And we know Lyme bacteria have that custom-fit extended UL23 loop.
So by understanding those distinct structural differences, medicinal chemists can move toward narrow-spectrum antibiotics.
They can tailor the chemical twists and bulky arms of a drug so that it only fits the tunnel of the specific pathogen they want to target.
Exactly. You could take a drug designed specifically to lock onto the unique loop of the Lyme bacteria in your system.
And because the healthy bacteria in your stomach don't have that specific loop,
the drug just passes right through them without binding.
It's highly targeted, personalized medicine driven by atomic-level architecture.
It really represents a massive leap forward. By understanding the minute structural quirks of each pathogen's machinery, we gain a significant advantage in staying ahead of their evolutionary resistance.
Yeah.
We are no longer just guessing, we're designing with precision.
That is just incredible. Okay, let's pull all of this together. For over 70 years, the medical community believed tetracyclines were relatively straightforward drugs. We thought they simply parked on the 30S subunit to jam the reading center of a bacterial factory.
Right.
But thanks to the transition from X-ray crystallography to the liquid state imaging of cryo-EM, we now know that is only half the story.
Only half.
These drugs are simultaneously navigating the 50S exit tunnel. They are contorting and flipping themselves to match the specific electrostatic environment of different pathogens.
In the case of drugs like Doxycycline, they're leveraging high local concentrations in the body to form multi-drug dimers.
Yeah, those massive trimers and dimers.
They build inescapable physical ceilings that block nascent proteins, and they actively wedge themselves into the machinery to disable folding and shipping processes.
Effectively bypassing the bacteria's attempts to mutate and resist.
Exactly.
It is just a remarkable mechanical process that has been happening inside millions of patients for decades, entirely unseen, which leaves me with a thought I haven't been able to shake since reading this paper.
Oh yeah? What's that?
Well, we have studied tetracyclines extensively since the 1940s. It is one of the most famous, commonly prescribed classes of antibiotics in human history. And we are just now, thanks to new imaging technology, seeing its secret secondary attack mode. So if a medicine this ubiquitous was hiding a massive dual-site mechanism right under our microscopes for nearly a century, what other ancient, everyday medicines are secretly operating in ways we haven't even begun to map?
That is a fascinating question.
Right. Next time you take a common prescription, it is worth asking yourself, what atomic-level mechanics are playing out inside your cells that science hasn't even discovered yet?
It's a great reminder that biology rarely settles for simple answers. There is always another layer of complexity waiting to be uncovered.
Absolutely. Well, thank you for joining us on this deep dive. Keep questioning the established dogma and keep exploring.